Gene expression and gene mutations. Gene mutations might arise during the replication of DNA. Any change to the sequence of bases or the rearrangement of bases within the DNA code is known as a gene mutation. Changes to one or more bases in the DNA triplets could lead to a change in the amino acid sequence of the polypeptide, the primary structure. Substitution of bases. The type of gene mutation in which a nucleotide in a section of a DNA molecule is replaced by another nucleotide that has a different base is known as substitution. Depending on which new base is substituted for the original, there are three possible consequences. So the formation of one of the three stop codons could occur, and that would mark the end of the polypeptide. As a result, the production of the polypeptide coded for by the section of DNA would be stopped prematurely. The final protein would almost certainly be significantly different, and the protein could not perform its normal function. A second option would be the formation of a codon for a different amino acid, meaning that the structure of the polypeptide produced would differ in a single amino acid. The protein of which this polypeptide is a part may differ in shape and not function properly. For example, if it is an enzyme, its active site may no longer fit the substrate and it will not catalyse the reaction, because the active site is a different shape and no longer complementary to the substrate shape. An example of this form of substitution mutation causes a condition called sickle cell anemia. The formation of a different codon, but one that produces a codon for the same amino acid as before. This is because the genetic code is degenerate, and so most amino acids have more than one codon. The mutation therefore has no effect on the polypeptide produced, and so the mutation is said to be a silent mutation. Deletion of bases. Minor though the loss of a single base might seem, the impact on the phenotype can be enormous. The one deleted base creates what is known as a frame shift, because the reading frame that contains each three letters of the code has been shifted to the left by one letter. The gene is now read in the wrong three base groups, and the coded information is altered. Most triplets will then be different, as will the amino acids they code for. The polypeptides will be different and lead to the production of a non-functional protein that could considerably alter the phenotype. One deleted base at the very start of a sequence could alter every triplet in the sequence. A deleted base near the end of the sequence is likely to have a smaller impact, but could still have consequences. An example of a deletion mutation is simply if you have a DNA sequence of TGCA and then you delete the G, it then becomes TCA, and this codes for a different amino acid. Other types of gene mutation. Addition of bases. If you add an extra base into the sequence, then you get a frame shift, and the whole sequence of bases becomes altered. The frame shift is to the right, not the left, as with deletion mutations. If you add 3 extra bases, or a multiple of 3, then there will not be a frame shift, but you may get an extra amino acid that was not previously included. Duplication of bases. One or more bases are repeated, and this produces a frame shift to the right. Inversion of bases. A group of bases becomes separate from the DNA sequence and rejoins at the same position, but in the inverse or back to front order. The base sequence of this portion is therefore reversed and affects the amino acid sequence as a result. Translocation of bases. A group of bases becomes separate from the DNA sequence on one chromosome and becomes inserted into the DNA sequence of a different chromosome. Translocations often have significant effects on gene expression, leading to an abnormal phenotype. These effects include the development of certain forms of cancer and also reduced fertility. Causes of mutation. Gene mutations can arise spontaneously during DNA replication. Spontaneous mutations are permanent changes in the DNA that occur without any outside influence. Despite being random occurrences, mutations occur with a predictable frequency. The natural mutation rate from species to species, but is typically around 1 or 2 mutations per 100,000 genes per generation. This basic mutation rate can be increased by outside factors known as mutagenic agents, and these include high energy ionizing radiation, for example alpha and beta particles as well as short wavelength radiation such as X-rays and ultraviolet light. These forms of radiation can disrupt the structure of the DNA. Chemicals, such as nitrogen dioxide, might directly alter the structure of DNA or interfere with transcription. Benzopyrene, a constituent of tobacco smoke, is a powerful mutagen that inactivates a tumour suppressor gene, TP53, leading to cancer. Mutations have both costs and benefits. On the one hand, they produce the genetic diversity necessary for natural selection and speciation, but on the other, they almost always give harmful and produce an organism that is less well suited to its environment. Additionally, mutations that occur in body cells rather than in gametes lead to disruption of normal cellular activities such as cell division, and this in turn could lead to cancer.